All right. No one is introducing me. I had hoped for David, sorry. To introduce me as another renowned scientist. I like those introductions. You're always very nice to our invited speakers. But I guess I'm just not good enough. That's fine. So let's go. It's okay. The other thing I will say, I will have to leave at 3:45 because I'm on a committee to select the new named professors in the college this year. Don't ask me why. I said yes, that was the reason. And so we'll go through this. And I also, the papers that I'll talk about, I will post those on Canvas so you can read those as well. Normally, actually, we discuss the papers, and then I just realized yesterday or so that, we didn't send out the papers, which is fine. But so what I thought I can do is just give a little bit of background on where this whole idea for this training ground came from, and to talk about two papers that I've written that are really close to the ideas of crossing scales. And the first one is a review I wrote together with two of my colleagues, Peter Johnson at the University of Colorado and Andy Fenton at the University of Liverpool. And this idea was really born during one of the Ecology and Evolution of Infectious Disease meetings, where we basically we're just chatting about community ecology and infectious disease, because we all study disease ecology. And I said, we really should think about how can we use community ecology to understand disease questions. And community ecology is a very broad field, but one of the most important things that we try to understand in community ecology is the different skills and how different skills affect each other. So in this review, what we did, this is my favorite figure from that review, and I spent a lot of time on this figure thinking about how the different levels and skills interact with each other. So here what we have is we have the different skills going from individual hosts, so we can really think about individual hosts being affected with pathogens. Of course, there can be multiple pathogens, so there can be pathogen communities within those individual hosts, but those pathogens who interact with each other, but also with the immune molecules and the tissues and the general physiology within those hosts. So you've got those level processes. And so this is typically when you think about pathology or immunology departments or graduate programs really thinking about that skill. How can we understand those interactions? Then we have the population level, and of course, being in the population biology, ecology, and evolution program, think a lot about populations. My degree was in population biology, so I have always thought a lot about what happens at the population scale. And of course, when we think about infectious diseases, a lot of the epidemiology that we think about happens in that population as well. So that population can be a local population, say in Atlanta or even this room. It can be a large population, even worldwide population, right? So, but generally speaking, we refer to a population as a group of individuals belonging to the same species. Now, then what we can do is think about how those populations then form communities. And so from the ecological point of view, communities are different populations of different species that are living in the same place, that may or may not interact with each other. But it's really when you go anywhere, of course, there's going to be multiple species. It's not just, in this case, humans. And then from those communities, we can think about scaling to regions and the whole world, so the global scale. And so what is important is that all these processes have originated, are often studied in isolation. So people may really say, I'm an immunologist, I'm here, I'm a, epidemiologist really looking at this. You could be, someone thinking about global health at the top there. And then disease ecologists often think about, things spilling over between species. You would be looking at that community level. But the point is that all of these levels, all these skills, the processes there will affect the other skills, and there's feedbacks, right? So it goes up and it goes down. And so what we did here was really thinking about, well, if that's the case, what have we learned from ecological theory and community ecology in general? And so we also thought about invasion ecology, and there's two approaches that are often used when we try to understand these processes. One is the offensive approach, and so when we think about invasion ecology, what we're trying to do is prevent dispersal from one place to another. And of course, the other direction, you would have a defensive approach, and we would say we're going to prevent the establishment. So it really depends on, you're here, does it go there, or does it come to you? That is basically the difference there. And so we were thinking about all these processes, how they affect each other. A lot of the paper that we wrote is about that basic community ecology, and this figure is really also about the applications. how can you start understanding infectious disease and controlling them by really thinking about these cross-scale dynamics. So for example, when we look at individual hosts, you can think about within host communities. And so when we take some of these defensive approaches, preventing the establishment of pathogens, we can think of recreating healthy microbiomes. So a great example here is C. difficile, was still called Clostridium, and he wrote his paper. It's now different. But it's really to stop, can we have a healthy microbiome to stop the invasion of pathogens like that? And there's a lot of other systems, for example, in mosquitoes, where people are introducing other symbionts, such as Wolbachia, to stop them from being good vectors for dengue or malaria, things like that. So that's really thinking about, in the individual host scale, how can we use these processes to control disease? So we can also think about how do you prevent things coming from different species to human populations, or from human populations to animal populations. So a lot of research now understanding also how we spread diseases to other species. It's not a one-way street. And there you can think about, okay, so if we think about vaccination, of course, dengue is a very big thing right now where vaccination is under trial. It's not going as well as people had hoped. But when people are really starting to think about dengue, what kind of vaccines do we produce, right? There is the cycles of dengue in human populations, but then there's the sylvatic cycle where the dengue viruses come from other primate species and actually jump to humans on a very regular basis. So if you're designing these vaccines, taking that into consideration, that you're not surprised every time you get that sylvatic that spill over into the human population. And then one thing that I really like is, when you start thinking about public health or conservation biology as a public health strategy, right? So we're thinking about creating these different, or preventing the transmission of particular viruses to human populations. And so preserving biodiversity can be a really effective way of diluting the risk of parasite infection. And in this case, so people are actually thinking about ways that used to be used, where you have a lot of, domestic animals can be used kind of as a sink for incoming pathogens so that we don't get infected as much. All right, so then the other way we can think of this is, so when we look at population levels, of course, heterogeneity is a really important part of all of this. And so a lot of us have heard of super spreaders, and super spreaders are interesting because we used to think of them as super spreaders. Now we think of them more as super spreading events. It's not necessarily biological reasons why some people are really good at spreading things, but it can be events, or it can be rituals or cultural things that people do. So for example, and this was really important during some of the Ebola outbreaks, as with the traditional funerals were really important aspects of transmitting the virus. So by implementing more sanitary burial practices, you can really take a lot of that heterogeneity away and really stop that transmission or really reduce it. Then when we think about reducing the abundance of disease in other species, right? So rabies is a really good example. So rabies is often coming from domestic dogs or other species. And so people have used bait vaccination. There's a lot of research going on in some places in Africa right now, where it's really trying to optimize how much bait do you need to put out to really stop that reservoir from bringing rabies back into the human population all the time. And that's an ecological problem. Spatial buffers. So when we think about scaling, there's also, of course, this is what Lance loves about scaling. It's like local and then spreading it out, right? And so, but you can have really important scaling at local scales, if you like. And so this is where people have thought about, for example, Nipah virus that comes from fruits through pigs to humans. And so a big part of that transmission cycle is actually the bats roosting in the fruit trees, the pigs feeding underneath, and then transmitting it to humans. Even creating the spatial barrier so that pigs don't go to the fruit trees can be very effective. And I know this is also the case with Hendra virus, where it goes from bats to horses to humans. I think we heard this in a talk recently in one of these seminars. So you can think of the same thing there as well. And of course, then we go from these communities to regions to the global scale. We can really think about how do we stop some of that transmission. Now, of course, when we had the COVID pandemic, we thought a lot about border closures. One of the things that I thought was really interesting, there's this fantastic book called Mosquito. It's about mosquitoes. And it's just like one of my favorite popular science books. But one of the things I learned from that is that the worldwide tire trade is a really important thing for spreading these disease vectors. And, I think a lot of us know when you have a tire, just a car tire, and rainwater gets in there, you can't get it out. So it's a perfect breeding ground for mosquitoes. And if these tires get loaded on ships and they get transported around the world, there's a whole worldwide network of these tires being transported. And they have actually been extremely important for the dispersal of Aedes albopictus. So even, I'm not going to say simple, I want to say simple, I know it's really difficult with trade and politics, but thinking about, what is actually creating these connections between these really distant areas around the world, you find out it's something as stupid as trade in tires. Think of drilling holes in these tires would already go a long way for the water to seep out, right? So sometimes solutions may be easier than we think if we think about how these skills are connected. And so rather than figuring out, okay, we're going to deal with these mosquitoes at this scale, so let's think about how do we stop them from coming here or taking them somewhere else in the first place. So to me, this figure has always been kind of the leading thread through our old training program, and versions of this figure have made it into our training grant proposal and renewal in our presentations as well. So that's really the big framework that I think a lot of what we're doing in this room comes back to. So that's paper one. Questions, discussion before I
thought you said worldwide tiger trade.
Tiger. Did I?
No, you said tiger, but I thought you said tiger.
Tiger.
Drilling holes in tiger.
Tiger mosquitoes. Well, I do have a subject.
No, I do think, we had a PBE dissertation a couple years ago looking at the standing water in boats going up and down the upper Amazon and how many mosquitoes are losing. Those are the storage areas, they rest on boxes and they, so. Yeah. So, and the thing that I'll add, the drilling a hole in the tires, that's not directly part of the disease process. So sometimes when we're looking at disease ecology, we're looking at every single piece and trying to intervene on that organism or that process. But sometimes it's something in between that can break the cycle. So we don't often think of the role of standing water, even though it's in, it's kind of in the life cycle. So I think there's a lot of public health interventions that aren't directly on the pathogen or on the vector or on the host that can be really effective, sometimes more effective than working on it. So I think that's something to keep in mind in precision medicine. If you're customizing your treatment to the genetics of an individual, whether that's a host animal or a person, sometimes that's a very expensive solution and may not be as effective as something. And we've seen that, we saw that in the presentation earlier.
Yeah.
So just bring that up.
No, and I like that about thinking about it in a framework, that's really developed for ecological understanding, not with the goal of we really need to, cure this particular species, right? But really trying to understand how all these skills are related to each other, how all these connections exist. And I like that example, but I also really like the idea of, hey, let's think of biodiversity, right? And really, maybe we can divert some of the money away that we spend on some medical interventions that may not be as effective as preserving forests. I really like that as an ecologist. I think that is a really cool idea. Cool. All right. So let me talk about next paper. And this is a more concrete example of how I've been thinking about within and between host processes. And so this is actually a project that we did with Mary Bushman, who was a PBE student here quite a few years ago now. She actually gave a talk in PBE 2 weeks ago-ish. And so this was in collaboration with Rustam Antia and also Kumar, who was at the CDC but retired a few years ago. But this was a really fun collaboration. This Kumar led the malaria unit at the CDC, and they basically had a lot of malaria samples from all around the world. So his main interest was in trying to understand drug resistance, how it evolves, how it changes, how it spreads across the globe. And they had freezers full of samples that they used for diagnostics, but that you could dive into and start asking ecological questions as well. So this was a really fun project that we were able to do. The project focused on Plasmodium falciparum, so there are multiple species of malaria parasites. This is the most devastating one, the most virulent, and the most common around the world. That's what we focused on. And it's a single-celled eukaryote, and it replicates in red blood cells in humans, and it's vector-borne. And it's always interesting to think about malaria parasites. You know, when you talk to parasitologists, they say we are the host and mosquitoes are the vectors. I like to think that the mosquitoes are the host and we are the vectors. And the reason I'm saying that is that the most important life stages, that is sexual reproduction, actually happens in the mosquitoes. I think it's much more interesting to think of us as a reservoir host and the mosquitoes as the real victims in this. Yeah, of course we don't care about mosquitoes like we do about humans, but it's always good to think about, you know, people used to think of these mosquitoes as just flying syringes, right? And a lot of textbooks still talk about them as if they're, you know, they don't do anything interesting to the parasites, which is not true. That said, I'm not going to talk about mosquitoes. I'm going to focus on the human part of this, but I think that was an important thing to mention. But we are looking at this from the human side of things, or certainly the mammalian side of things. So when we think about malaria, there's a lot of anti-malaria drug resistance, and this is just one example of many different maps I could have shown here. This is chloroquine sensitivity in the yellowish light green, and then resistant parasites that you find in all these different regions around the world. So very, very widespread. Now this is chloroquine. This is still a drug that gets used. It's not the main line, right? Like artemisinin, which is right now. But it has been used a lot historically, and it's still used in a lot of different places, but there is a lot of resistance, and that is why people are looking for other options to use. Now, one of the most surprising things about drug resistance in malaria, and this isn't just true for chloroquine, this is also true for other forms of resistance to other drugs, is that you think about where is malaria most common, where is it most prevalent. Sub-Saharan Africa is, if you want to get malaria, you go to Sub-Saharan Africa, right? If you want to be less likely to get it, you know, you go to Asia or South America. So what's interesting though is that resistance didn't actually evolve in Sub-Saharan Africa. So what population genetics tell us when we look at the mutations involved in drug resistance in malaria is that they first evolved either in Southeast Asia, and you see the times that chloroquine resistance actually detected, this was all in the 50s and 60s, it was only in 1978 that it was detected in Africa. You see it earlier in South America. And this is true for other forms of malaria resistance too. It never starts in Sub-Saharan Africa, but when it gets there, it spreads like wildfire, right? So intuitively, you say, well, where's the selection pressure the highest? Must be Africa, because that's where most malaria is, that's where most drug resistance happens, or drug treatment happens. That's where you would expect most selection. It's not what we see. We see it spreading once it gets there, but we don't see it originating there. So this is a really interesting ecological puzzle, because it's like, why doesn't it happen there? What's going on there? So our thesis is that what happens within hosts, interactions between parasites is explaining this pattern. Okay, so when we think about malaria infections, what we often find is that individuals are infected with multiple strains of the parasite, not just one, and so we refer to that as multiplicity of infection, the number of clones or strains, however you want to call them, inside a host. And then other important factors are transmission intensity, so you have way more transmission, more infected mosquitoes flying around, you get more infectious bites. We see that, of course, the frequency of those infections bites goes up, but this also results in higher levels of multiplicity of infection, which makes sense, right? We get more bites from infected mosquitoes, you're more likely to be infected with multiple strains of malaria. And that is what we see in human populations. So when we think about this multiplicity of infection, the question then is, can we think about interactions between drug-resistant and sensitive strains, competition between them, a very basic ecological process? Can it explain how we see this transmission or this occurrence of resistance having a low transmission area and not a high transmission area? And a lot of this work started during my PhD. So this was one of my papers published in 2004 in the Malaria Journal. I think this was the first year that journal existed. maybe second year, I don't know. But so I did my PhD on rodent malaria parasites that we looked at in mice. You may ask, why am I working on monarch butterflies now? The reason is that it was really interesting to work on this, but it wasn't a natural system, right? We used Plasmodium schiavoni, which is a parasite that normally occurs in thicket rats. We do it in lab mice. Not very natural, right? You can use it for some things, but ultimately I wanted to work on a parasite in a natural host that I can do field work on and experiments. And that's why I work on devastating parasites in monarch butterflies right now. But what we did here is my whole thesis was about understanding within host competition. And so what we have here, two strains of malaria parasite, resistant and sensitive. We don't show the sensitive here. So I'm just showing the resistant parasite on its own in solid lines and the resistant parasite in mixed infections in dashed lines. This was before we did all our publications in beautiful colors. You know, we still had to be able to print it in black and white and still make it visible. So, but here we have the solid line. So this is an untreated mice. You can see when the parasite is on its own, reaches a really high peak, comes back, the overall stays at high levels. And this is a log scale, right? So these are big differences. And then the dashed line is when the parasite is in the same host as a sensitive parasite, way fewer of them. When you then drug treat the mice, there's no difference, right? So you drug treat the mice and then the resistant parasites do as well, with or without the sensitive parasite. Makes sense because the sensitive one gets killed, the resistant one grows up, right? So this is demonstrating ecological competition. Sensitive strains suppress the resistant strains. All right, so is this true in humans too? So this was the first question that Mary addressed in her thesis. So after I moved to monarch butterflies and then came to Emory, I was still interested in malaria, but I wanted to study human malaria. It's way harder than mouse malaria, but also more relevant in some ways. So this is when we started a collaboration with Kumar and the CDC. And so what we had blood samples from infected children that were collected before treatment. And then we looked specifically at mutations in the PFCRT gene, which is involved in resistance to chloroquine. And we used quantitative real-time PCR to look at the density of the resistant and the sensitive strains. And so we're specifically looking at we were defining these strains based on the resistance or that chloroquine resistance gene. There could be other differences, right? So it could be a pool of different genotypes, but we're specifically looking at the populations that are resistant versus sensitive to this drug. And we looked at 3 different populations. In fact, and this was kind of interesting, we started with this data set from Ghana, where we had 680 samples. And what is nice is we have different levels of transmission intensity. So the redder, the more transmission, the bluer, the less transmission. And when we got our results, we're super excited. But sometimes when you're excited about your results, you get worried that it is a fluke. So we did it again in Tanzania, and then it showed the same. And we said, I'm still not convinced. So we did it in Angola too, and we found the same results in all 3 studies, which we were more confident at that point that things made sense. All right, so the first thing that we can do is look at the overall parasite density in these samples. And this is as a function of the number of strains that we could detect. And this is based on microsatellites, not the resistance or sensitivity genotype. But the important thing here is, the more different parasites there are, you don't see an increase in overall parasite density, right? So that shows us there is a carrying capacity in the bloodstream of these children. And if there's more strains, they have to divide the available numbers of parasites between them. That's ecological competition. So this is demonstrating, yes, there is competition. So now we can ask who is actually suffering from this competition. So what I'm showing here is when we have the chloroquine, this is all about chloroquine sensitive parasites, when they're on their own in this kind of toothpaste color, and then when they're in infections with the resistant parasite in the kind of baby blue color. And what we can see is that in all these 3 countries, in all these samples, we see that these parasites do worse when there is resistant parasites around, right? So sensitive parasites are suppressed by resistant parasites. And we see the opposite too. The resistant parasites, when they are in the presence of the resistant parasites, that's the dark red, they do worse, they have lower densities than when they're on their own in this kind of pinkish color. So what this is showing us is that there are similar things that we see in mice, where we can control everything and track everything over time. We can see this in a cross-sectional survey across 3 different countries with real samples from children. You can see that there is this competition. And when these parasites have to share a host, they do worse. They're competitively suppressed. Now we can also look at, when we just look at the overall densities of these parasites, what we find is that when we look at these single genotype infections, we also find that the resistant ones, in some cases, do worse on their own than the sensitive parasite. And this indicates that there is some cost of resistance, right? And this is often found in lots of different systems. When you're resistant, you pay the price for resistance. This is true for mosquitoes, it's true for malaria parasites, it's for a lot of other things as well. So there's some fitness cost associated with carrying that resistance. And you see that in this case by finding that the resistant strains, even when they're on their own, they don't do as well as the sensitive strains would do on their own. And then what we did is we did a very simple model where we say, well, this is what we would expect just based on the frequencies of the parasites in the population, in the green bars, the purple bars. And so this is the fraction within individuals, like the proportion of the parasite population that consists of resistant parasites. The purple is what we actually observe. So we find that we observe less than we would expect if there was no fitness cost. So this indicates that fitness cost of resistance. Now in the Ghana data set, we could also look over time at chloroquine resistance. So this is the proportion of infections that were positive for the resistance gene. In 2004, chloroquine was retired as a first-line drug treatment in Ghana, and what we can see is following that time, there is a decline in the frequency of the gene, right? So consistent with natural selection, selecting against this resistance gene. All right, any questions so far before I dive into the model connecting all of this good to population level processes? No? Good. All right. All right, so what we did next was then to say, okay, so we know this competition happens. And so here is really the hypothesis, right? So when you think again about that big scale pattern that we saw, we go to sub-Saharan Africa where there's tons of transmission. That means there's lots of infections, there's a lot of drug treatment, but we don't see resistance first happening there. So could it be that because there's so much infection, most infections are consisting of multiple strains, and if there's this fitness cost and you're competitively suppressed, you can evolve resistance, but there's nowhere to go. You're suppressed, you cannot spread. That's really the hypothesis that we were testing here. And the way we were testing that is with a modeling approach. So we did within host dynamics using differential equations. And this is it's nice because we have so much research that comes from the mouse malaria literature, but also from the unethical literature where people were treated with malaria parasites to treat syphilis, right, to get fevers up, and that would then treat syphilis. And so the CDC has kept data of that where we actually can understand the within host dynamics of human malaria infections as experiments as well. So we can look at asexual parasites, the gametocytes, which are the ones that actually make it to mosquitoes. And we can also look at innate and acquired immunity. And all of that work is really based on all the work that Rustem has done, really trying to understand how immunity works as a quantitative process. And then for the between host dynamics, we did an individual-based model, also known as an agent-based model, an ABM, as they're often called. And so what we had there is 400 humans in a population, 12,000 mosquitoes, and we would run this for many years. So basically, what we're doing is all those individual humans and mosquitoes, we keep track of their individual identity. We start with a number of infected individuals in this population. They go through their processes. They produce gametocytes. Those go to mosquitoes. We keep track of the individual mosquito's lifetime. If they make it to 2 weeks old and they bite someone, there's a certain probability that they infect that person. If they die beforehand, which can happen, they don't. And so it takes a lot of computing power, and you run simulations and you run them over and over again to try to understand what our general process is, right? So we have our differential equations, which are really, deterministic. They're going to tell you what would happen given certain conditions. But then the ABM is very much stochastic. You run it again, the result will look different, right? So, and so that's what we did. So to start with the within host dynamics, the first important thing was to really see if we can regenerate the dynamics that we know should happen in malaria infections. And so what we're looking at here, I start with the idea that there's no fitness costs at all. And in blue here, so what we're doing here is we start an infection with a sensitive parasite, and we're infecting one of these humans in this model. And what we see is there's a big spike that comes down, goes up, comes down, and it kind of eventually goes down. And this is very much what we see with real malaria infections. So malaria parasites have antigenic variation, right? So they grow up, but then the parasites change their antigenic properties throughout the infection. So you get the immune system developing antibodies to specific, antigenic properties on the parasites, but they change them so they can escape the immune response. The immune system has to catch up, and so you get these chronic infections. But overall, in the end, the parasite infection goes down. But you see that almost takes a year, right? So this is why you get these really long chronic infections of malaria in human populations. Now in red here is the resistant parasite, and you can hardly see it. So we introduced it after the sensitive parasite was in there. This is showing that the model shows us competition. Once there's already a parasite, they're really hard to get in, especially when there's high cross-reactivity. So this means that the immune system, the innate immune response, is very likely to affect both parasites, right? So it's elicited by the sensitive parasite in this case. It means it's also going to act against the resistant parasite, very much so. All right, if we bring in the resistant parasite a little bit later, when most of the immunity is much more specific, based on antibodies specific to the sensitive parasites, less based on the innate immunity, we can see the parasite actually coming up a little bit. I mean, still, it's on a log scale, so it is way lower. But you can see the effect of the different immunity, innate versus acquired immunity, playing out here. Now, if there's low cross-reactivity, you get much more interesting stuff. So here you start with the sensitive parasite, you bring in the resistant. The resistant parasite could actually take over if there is low cross-reactivity. And that means if there's less competition mediated through the immune system. And we also see that if you introduce the parasite later. Right, so these are all dynamics that we, actually a lot of the work that I did with mice was finding things like this in mice. So this is very similar to what we found in mice. And it's consistent with the broad scale dynamics we see in human infections as well. All right, so now what we can do is we can add a fitness cost. And so here we're actually looking at single infections of the resistant and sensitive parasite. And it's hard to see, but the resistant parasites don't do as well. But that cost is way amplified when both parasites are there. So in a mixed infection, that 10% fitness cost, just saying you have 10% lower fitness when you're resistant, is really hammering you in that competition with the sensitive parasites. Again, these are log scales, right? So there's orders of magnitude difference there. So what that's telling us is that competition can really amplify the cost of resistance. And so these resistant parasites don't do well. Now we look at the population level dynamics. What we can see, so this is the bites per host per day. So from 5 to 20, which is kind of consistent with some of the transmission intensities that we see in natural populations. What we can see is that the more bites you get, the more infection there is in the population. Makes perfect sense. Still good to know that the model works, right? So these are all the things you want to test. The frequency of mixed infections also goes up. That makes sense as well. More bites means more mixed strain infections. And this is also in line with a lot of epidemiological studies that have shown higher transmission areas get more mixed infections. What is especially nice is that there is also this pattern of age-dependent immunity and resistance in malaria populations. So what we find is when you go to a high transmission population, you find that most infections are in the youngest children in that population. If you go to a low transmission area, so say you go to a place in Southeast Asia, you actually find that the relative numbers of younger and older people infected are much more similar, right? So you get these way more skewed distributions in high transmission areas, and our model shows that. So this is consistent with what we see for high transmission. This is consistent with what we see in real populations for low transmission. So this agent-based model is really recapitulating a lot of the things that we see that characterize these natural infections in human populations. All right, so having shown that both the within host dynamics and the population level processes are consistent with what we know about malaria, we can then start the simulations to see, okay, so now we have our population. Let's say we start with a population that only has sensitive parasites, and now I'm going to bring in a resistant parasite, right? So this would be the way that evolution works. You get mutations to give rise to resistance. Now you're going to see, can they take off in that population? Can it actually happen? All right, so we start with no fitness cost and no selection. So this would be a population where there's basically no drug usage, right? So, okay, so a low transmission area, where we see this is the number of infected individuals over time. We get this over 12 years, so it's a lot of simulation. In blue is the sensitive parasite. We introduce the resistant parasite in red. You can see, this variation is based on the different runs, but it's hardly making it in there. And so in black also is the infections that have both types of parasites. High transmission, you get more people infected. So the blue line is higher, that makes sense. But the red doesn't even have a chance, right? So in that high transmission where you have a lot of mixed infections, you get a new parasite, in this case it's resistant, there's no way. It just cannot win that competition, actually get into this population. Then we can look at, so this was all for high cross-reactivity. That means, again, that the immune system affects both of them much more similarly. That means stronger competition. We can relieve that a little bit, have low cross-reactivity. And then you can see that there is this competitive release. So this is where this new parasite that comes in red can actually start increasing in frequency, because even if the other parasite is already there, it can come in because the immune system doesn't suppress it as soon as it comes in. Right? So lower competition, there is more chance of the parasite to come in. But the increased transmission, there's more of these mixed infections, and the parasite, again, hardly has any chance of getting in. All right, so but this is without drugs, right? So you get a resistant parasite, it doesn't really have a fitness advantage. It doesn't really go anywhere, especially when there is high transmission. All right, but now we're going to introduce some drugs into the population, because that's, of course, what we're interested in. And so if we start with a low transmission area, and this is actually showing high cross-reactivity, so you would expect a lot of competition, but you see the sensitive parasites go down, and this new parasite that's resistant, that's being selected for because there is drug usage, it goes up over time. So it can invade this population. And then when it's high transmission, it can also invade it, but you also see tons of variation, right? These are basically error bars of our different, so this means these different populations do all sorts of different things over time. But you can get this here when you have this moderate selection, when you have drug use in the population. All right, and then if we look at low cross-reactivity here, again, the parasite can come in, and then with our high transmission, it's doing really well. So there's less competition, lots of selection. That's when the resistant parasite comes in. But that's not what we find, right? Because we know these fitness costs exist, and this has actually been shown in our own paper. A lot of our papers show resistance is not cost-free for the parasite. So let's add the fitness cost, and we still have moderate selection, so we're throwing drugs at people in this population, but there is a fitness cost. Now, in low transmission, you can get this resistant parasite, can overcome that cost and actually take off in the population. But this is the interesting part, where this is when we look at sub-Saharan Africa. This is where we have our sensitive parasite. We have drug use, but there's a cost to resistance. There's high transmission, meaning that most of the infections are consisting of multiple strains. So in this case, the resistant parasite just cannot get into the population. So the cost of its resistance is exacerbated by this competition, and it's not going to take off. So really, this is very much the pattern that we see, right? So this would be like South Africa, or South America, or Southeast Asia. This would be more like sub-Saharan Africa. And so when we look at lower cross-reactivity, we see the patterns are a little bit different, but the same pattern holds, that when we high transmission, that resistant parasite doesn't really get in very easily at first. Okay, but now we're gonna do the strong selection. right? So this was like moderate, but this strong selection would be treating everyone, like mass drug campaigns as are being proposed or done in certain places. And now we see, the resistant parasite comes in, even when there's high transmission, if you throw enough drugs at them, they do well. And so that's what we're seeing. So what all of this is telling us is that when you think about the initial situations of drug resistance, when the parasites, when you look at a high transmission area, you get the evolution of a drug resistant strain. At that point, there's not massive drug treatment. There may be some, so there's some selection, but there's a cost to resistance. The parasites suffer from this competition, they cannot take off. But then later on, you get to a point where there is resistance and people start using tons and tons of drugs, and you see the rate of increase is very, very rapid. So what this is telling us is that there's basically two things to drug resistance. There is the initial emergence, and then there's the subsequent spread. And so what we're seeing is that for a drug resistant strain to emerge and first take off, what is best for them is a low transmission area, because there's less competition within hosts, therefore it can spread to more people in the population. But once it's established and there's a lot of drugs, then the high transmission actually helps them because they can transmit it to more and more people, right? So in a way, really, this tells us that what happens within host is these interactions between parasites within a host can really have really important aspects at the population level. But what happens at the population level feeds back into that, right? But that higher transmission then also means that the resistant parasites can transmit more. And so I think what this paper told us is that we have this emergence and spread. Those are two different things. You need to understand what happens within host and the population level to try to understand some of these patterns that have really been puzzling, right? And so it's kind of nice because you look at the global scale, you see these weird patterns. I expect drug resistance to originate there. It doesn't. It originates there. Once it gets there, it goes through the roof, right? So you have a global scale, population level, within host level, and they can all interact together to really explain these different patterns. So with that, I thank you. And I have to run to this other meeting. But I would be very happy to answer any questions you may have by email or our next meeting.